Hello, welcome to Lead Lying Down. I'm Tom Neer, ichthyologist, professor of ecology and evolutionary biology, and Bingham Oceanographic curator at the Yale Peabody Museum, all at Yale University. Well, I'm excited today because largemouth bass is in the news. As you probably know, largemouth bass is among the world's most popular freshwater sport fishes. This popularity has a drawback in that largemouth bass have been introduced all over the planet. So while only found in the eastern part of North America, extending a little bit into Mexico and Canada, as we could see from the map figure here, largemouth bass have been introduced all over the North American continent, all over Europe, South Africa, parts of Asia, and New Zealand. So in many parts of the world, albeit a popular freshwater sport fish, largemouth bass is a nuisance and an invasive species. But that's not why largemouth bass are in the news. Largemouth bass are in the news because last week, the Florida Fish and Wildlife Conservation Commission announced that largemouth bass in Florida are now known as Florida bass. And this has resulted in the past week of a flood of news stories. By my count, more than 30 news stories that range from largemouth bass, now known as Florida bass, to goodbye largemouth bass. Florida's popular game fish has a new name. So this leads us to ask, what is in a name? And why the Florida bass is recognized as different from the largemouth bass. And this goes back to a research project published in my lab 2 years ago in June of 2022. This was a dissertation chapter of Dr. Damon Kim, who's just finished his 1-year postdoc in my lab after finishing his PhD and is heading to do a postdoctoral fellowship with Professor Rachel Moran at Texas A&M and Professor Andrew Taylor at the University of North Georgia. The three of us collaborated on a project where utilizing DNA sequences sampled across the genome of all the recognized species of black basses, genus Micropterus, which includes largemouth bass, smallmouth bass, and spotted bass, as well as 11 other species, to figure out their evolutionary relationships, their divergence times, and ask, does genomic data support the recognition of each of the species, and in particular focusing in on the largemouth and Florida bass. Now, there's really interesting areas of research in the biodiversity of black basses, the genus Micropterus. There are currently 14 species that are recognized. Nearly half of these, 6 of these species, have been described or elevated out of synonymy or elevated from subspecies status in the 21st century. So almost half of the living diversity of black basses that we see today have really come into the scientific dialogue in the last 20 years. So examples here include the Neosho bass, closely related to the smallmouth bass, as well as the Chattahoochee bass that's closely related to the red-eye bass in the Mobile Basin of Alabama. As many as 5 species of black basses are known. This is in addition to those 14 species that are known but undescribed species of Micropterus. This includes the Choctaw bass and Bartram's bass. But where do these bass species names come from? And in particular, where does the scientific name of the largemouth bass and the Florida bass come from? Well, for the largemouth bass has historically been classified as Micropterus salmoides. And we have to go way back to the earliest days of the 19th century in 1802, where the French biologist Lacépède presented a scientific description of Micropterus salmoides based on specimens that were collected in Charleston, South Carolina. And taxonomists refer to this as the type locality. Type specimens are specimens that are used by the taxonomists to describe the species, and they're set there as to provide an example of what that species is. So for Micropterus salmoides, that traditionally has been classified as the largemouth, called the largemouth bass, has been classified as Micropterus salmoides. The type locality is Charleston, South Carolina. The Florida bass was described only 20 years later with a type locality in eastern Florida by a French scientist who did a lot of work in North America, Lesueur. Now, in a monograph published in 1849 that we're going to talk about in a little bit of detail in this episode of Lead Lying Down, Reeve Bailey and Carl Hubbs state that although stated originally described from East Florida, it should be assumed that specimens that Lesueur based his description for Florida bass, Micropterus floridanus, came from the St. Johns River system in northeastern Florida, marked here with a star and an arrow. So you could see the St. Johns River outlet and Charleston, South Carolina, as the respective type localities for Micropterus selmoides that we see in Charleston, South Carolina, and Micropterus floridanus that we see in the St. Johns River, are very close to each other in terms of their spacing. Now, a monograph was published in 1949 by two giants in North American ichthyology, working at the time out of the University of Michigan, Reeve Bailey and his mentor Carl Hubbs. Now, the data that Bailey and Hubbs were using to delimit their species of basses were traits that ichthyologists have been using since the early 1800s. These are features that are counted in whole numbers. We call meuristics. So, for example, the number of lateral line scales along the fish, the number of dorsal spines in the first dorsal fin, the number of rays in the second dorsal fin, the number of rays in the anal fin and pectoral fin, has been noted by biologists going way back to the early 19th century that closely related species tend to differ consistently in these traits. So Bailey and Hubbs collected a large data set of these meuristic traits and delimited populations that they designated as largemouth bass and smallmouth bass, or Florida bass. And if we look at the map of the natural native populations of largemouth and Florida bass, Bailey and Hubbs noted that populations that had high counts, high meuristics, were limited to peninsular Florida, that they called a subspecies Micropterus selmoides floridanus. And populations with low scale counts, shown here in blue, throughout much of eastern North America, had low counts that they designated as subspecies Micropterus selmoides. Why did they call these different subspecies? Well, they called these different subspecies because they assumed that they were hybridizing. In the areas of the Gulf draining tributaries of the Florida Panhandle and extreme eastern Alabama, as well as the Gulf draining tributaries of Georgia and the Atlantic draining tributaries of Georgia and part of South Carolina up to the Savannah River system, they identified specimens that were intermediate in the counts. They weren't low like we see in blue largemouth bass. They weren't high like we see in yellow Florida bass. They were intermediate. And they presumed that this represented contact subsequent to their divergence where these hybrids are formed, the hybrids being intermediate. Now, Bailey and Hubbs were working at a time when biologists felt that if populations or lineages exhibited hybrids, they couldn't be different species because the definition of species were those lineages that were reproductively isolated from one another. What we know since that time, particularly with the onset of phylogenetic systematics, and when phylogenetic systematics entered thinking of species concepts, a different concept of species has been modified, the evolutionary lineage concept of species. That is, lineages may have their own distinct evolutionary trajectories. And if they do exhibit the ability to hybridize with other species, those could simply be an ancestral trait that hasn't been lost in the subsequent lineages. So Bailey and Hubbs, focusing on the biological species concept, treated these as subspecies, but yet treated these as different entities with a wide area of integration throughout much of the state of Georgia. And this is illustrated here in this table of their lateral line scale counts, where we see in the bottom row there in blue, we're going along the Atlantic coast, past the Savannah River, to Charleston, South Carolina, where we see on that bottom line, those populations have low counts that they think are consistent with largemouth bass. The top rows that are colored in blue represent those populations going from the Gulf Coast into the interior of Eastern North America, the Mississippi River system in particular, where those low counts are designated as largemouth bass. The yellow line right there in the middle represents populations measured from peninsular Florida, that they argued those high counts are consistent with pure Florida bass. And we have the populations at the second and third row, that is populations going on the from the Atlantic drainages from peninsular Florida and the Gulf Coast drainages from peninsular Florida, that we have these intermediate populations that they argued represent hybrids between these two, what they called subspecies. But yet, based on the meuristics, they noted that largemouth bass and Florida bass were distinct relative to one another. And in particular, looking at this area of intergradation, that is the area of the map that's bounded by this amorphous green line. What we see on the left with those frequency histograms in the bottom left part of the figure, that red line shows the demarcation between what they would argue is largemouth bass on the left, Florida bass on the right, as we're going from the peninsula along the Gulf Coast. And then the frequency histograms on the right side of the figure, going from peninsular Florida up the Atlantic coast. If we're distributed right of that red line, we're pure Florida bass. If those populations modally are on that line, those are the intergrades. And if the mode is well left of that line, those then were pure largemouth bass. This included the type locality for Micropterus salmoides in Charleston, South Carolina. Given the meuristics and the traits emphasized by Bailey and Hubbs, what is found in Charleston, South Carolina is largemouth bass, Micropterus salmoides. And this just outlines the traits that Bailey and Hubbs emphasized distinguishing largemouth and Florida bass. I just want to point out the last of those traits listed is that in largemouth bass, that mid-lateral stripe is not broken up into blotches, whereas in Florida bass, the anterior mid-lateral stripe, as you can see, is broken up into blotches. And this pigmentation was so consistent, but yet showed a geographic pattern that Bailey and Hubbs noted was against the pattern they found in the meuristics. What they were saying is that specimens from Atlantic coast drainages going up into low meuristic area where we expect largemouth bass to occur, these quote unquote largemouth bass had a pigmentation that was more consistent with what they saw in fishes in peninsular Florida, that is pure Florida bass. So Bailey and Hubbs are going all in with the meuristics, and they're treating the pigmentation in a sense as a parameter, as a trait that is not consistent with the delimitation that they have between largemouth and Florida bass. It's been nearly 75 years since Bailey and Hubbs' work, and since then, there have been very few studies that have challenged its conclusions. The first of which was a study based on protein allozymes published in 1983, and then two studies published in 2002 and 2003 using a combination of morphology, allozymes, and mitochondrial DNA, including divergence time estimates as well as phylogenetic estimates of phylogenetic relationships among the species of black basses. The study Kasser et al. 2002, and the study that I was the lead author on in 2003, we collaborated together on these papers. We proposed the Florida bass should be treated as a distinct species relative to the largemouth bass. Albeit, the American Fisheries Society Names Committee disagreed. And in the publication of their names list in 2004, they deferred formal recognition of Florida bass as a distinct species pending further work. And then 9 years later in 2013, they again did not accept our proposal that Florida bass is a distinct species relative to largemouth bass because no study from their perspective included populations from this intergrade zone. So while the biological species concept's use as an operational concept to delimit species by 2013 was out of date, the AFS Names Committee was still relying on a biological species concept with regard to justifying not adopting the proposal that largemouth bass and Florida bass are distinct species. So Dr. Damon Kim, Professor Andrew Taylor, and I decided to revisit the question using a phylogenomic analysis. That is a phylogenetic analysis of DNA sequences sampled across the genome. So here briefly is the methods we used. And in our phylogenetic analysis, indeed, we resolved two distinct lineages that correspond to Florida bass and largemouth bass. However, there was a huge surprise in these results. In particular, in the orange box includes populations of largemouth bass that from the Atlantic tributaries north of the Savannah River, including the type locality for Micropterus selmoides in Charleston, South Carolina. These populations are resolving as Florida bass. And indeed, if we redraw the map to reflect the delimitation that we're getting from the phylogenomic analysis relative to what Hubbs and Bailey proposed, what we see is that Florida bass genotypes extend all the way up to the Tar River system near the North Carolina-Virginia state line, and indeed include populations at the type locality of Micropterus selmoides. Therefore, Micropterus selmoides now refers to the Florida bass. Yes, this freaked us out as well. So we now have to ask if Micropterus selmoides is the appropriate name for the Florida bass, what is the available scientific name for the largemouth bass? So I have listed here are all of the published scientific names extending from the early to the mid-19th century for populations of what we know are now largemouth and Florida bass. So Micropterus selmoides is the oldest name, type locality at Charleston, South Carolina. That becomes the Florida bass. Micropterus floridanus is the next available name, but we know that those populations at the St. Johns River indeed are Florida bass, so that name becomes a synonym of Micropterus selmoides. The next available name that would apply to a population that we are delimiting as largemouth bass is Micropterus nigricans from 1822 by the Legends of Ichthyology, Cuvier and Valescens, who in their multi-volume work extending from the 1820s to the 1840s had an ambition of describing every species of fish on the planet. Well, here is another one they got right. The available scientific name for the largemouth bass is Micropterus nigricans, and we argue that Micropterus nigricans is the appropriate scientific name for largemouth bass. Micropterus selmoides no longer refers to the largemouth bass. That refers to the Florida bass with the type locality there at Charleston, South Carolina. The next available name is Micropterus nigricans, which is the appropriate scientific name for the largemouth bass with a type locality in Lake Huron. And in fact, while the species description is brief, we could see an artist's rendition of a specimen of Micropterus nigricans. Looks a lot like a largemouth bass to me. And given the description of the morphological traits in the description, and indeed it matches what we expect a largemouth bass morphological traits for that to exhibit. So selmoides becomes the scientific name for Florida bass. Micropterus nigricans becomes the scientific name for largemouth bass. And there are at least 3 synonyms, that is names proposed for species that we know are largemouth bass that were published after the description of Micropterus nigricans in 1822. Now, other lines of evidence for the distinctiveness of Florida and largemouth bass, we could look at genomic divergence. So we looked at pairwise FST. F statistics looks at the proportion of total alleles that in between the two species, the proportion that are shared relative to the total number of alleles at a locus. FST is a unitless measure, ranges from 0 to 1. 0 means complete sharing of alleles. 1 means complete non-sharing of alleles. As a general metric, it's expected that species would have an FST of 0.2 or 0.3 and above. And indeed, what we see is when we compare Micropterus nigricans and Micropterus salmoides, FSTs range from about 0.4 all the way past 0.6. And the FST we see in these species is far lower than we see when comparing populations within species and even comparing populations of nigricans and floridanus to the hybrids we found in the populations of Florida and largemouth bass. Now, with regard to hybridizations, we don't find that whole area of Georgia, Gulf draining tributaries, Atlantic draining tributaries are characterized by intergrades or hybrids. We think there's one area of natural hybridization in the western panhandle of Florida, the Choctawhatchee River. That's the only area we think natural hybrids occur between Florida and largemouth bass. And that large green swath we see there in Georgia and the Carolinas, those are up in the Piedmont areas, the upper stretches of rivers that drain those regions into the Atlantic Ocean. Those are areas where impoundments have been put in place, artificial lakes. And we think that naturally populations in those upper reaches of the rivers were Florida bass, Micropterus salmoides. And after the creation of these reservoirs, there was a steady input of stocking of largemouth bass from areas outside here. And given this introduction, we now have the non-natural hybridization between the two species that were brought together due to human activity. So hybridization is not as extensive as the meristics say, and perhaps that pigmentation pattern that Bailey and Hubbs noted was a more accurate delimiter of the species than were the meristics. The two species, largemouth bass and Florida bass, exhibit an ancient divergence time. So I'm showing you here as a phylogeny based on the genomic data that's scaled to the estimated times of evolutionary divergence, shown on the x-axis of the phylogeny. And in the orange box, I have the split between the phylogeny between largemouth and Florida bass, and that age is estimated at 2.3 million years. And that red dashed line cuts at 2.3 million years across the phylogeny. We can see that every other sister species pairs within Micropterus are younger than the divergence time that we estimate between largemouth bass and Florida bass. So Florida bass is a distinct species. If you don't believe us, let's turn to the American Fisheries Society names list, where just last year in 2023, they say that Micropterus nigricans removed from synonymy of Micropterus salmoides by Kim et al. 2022. That was our study. Kim et al. Recommended use of largemouth bass for this species and Florida bass for Micropterus salmoides, which we follow. So hurrah! The Florida bass is a distinct species. So DNA sequences from more than 51,000 genes spread across the nuclear genome provides a fully resolved phylogeny for the black basses. And this genomic evidence, along with morphological divergence as reflected in pigmentation, allows the delimitation of Florida bass and largemouth bass, resulting in an appropriate change of the scientific names for these species. Now, this new delimitation is not meant to confuse anyone. It's not meant to cause taxonomic chaos, but it's where we want the taxonomy to accurately reflect the science. We argue that after 75 years, it now does that. And we're providing a more accurate perspective for effective management and biological conservation. I have to say that as a taxonomist and evolutionary biologist, it gives me great pleasure to see taxonomic work becoming such big news in the 21st century. And it just shows that how we resolve the tree of life and the names we put at the tree of life, whether it's the most inclusive groups of the tree of life or the species at the tips, have real world implications for humanity's place in biodiversity. Thank you for your attention, and I look forward to seeing you again in the next Lead Line Down video. Goodbye.